Hello, AP Bio. Today we are going on to the next part of the process of protein synthesis. Today we're actually kind of in between. We did transcription yesterday, but if you remember where we left off, our messenger RNA strand that we made was not totally finished. It was what we call a pre-mRNA. So today we're going to talk about how the messenger RNA kind of gets finished off. And then we're going to start talking about the pieces and the molecules that are involved in the second half, the process of translation. And so there are a couple different things we're going to talk about today as we move from one to the other. You will need the messenger RNA strand that you built yesterday. The DNA is still here just in case you need to double check. We're not going to use this very much today, but this is also just to remind us that we're still in the nucleus with the DNA at the beginning today. You should have had several adenine bases left over. We'll need to use a few of those today. And today we will also need your GTP cap, this little tiny piece that was stapled in your packet. You will also need some extra paper. It could be construction paper or notebook paper, but we're going to be making one other piece. We'll be making a ribosome to use as part of this modeling activity also. So as you get all of those pieces, take a minute and think about what you did yesterday and see if you can use this vocabulary to explain the process of transcription. So once the primary transcript or that pre-mRNA has been made, it needs some modifications before it can exit the nucleus and head out to a ribosome. So it's not quite finished. The 5' end of the messenger RNA receives what's called a 5' cap. It's a modified guanine nucleotide, guanine triphosphate, so GTP, that we're going to put on the 5' end, and the 3' end receives a poly A tail, which is actually 50 to 250 more adenine nucleotides tagged onto the 3' end. The purpose of these is that they help export the messenger RNA out of the nucleus. They give the proteins that help move it out of the nucleus something to grab onto. They also help protect it so that it's not broken down before it can be translated into a protein. And it helps the ribosome figure out where to attach once the messenger RNA travels out to the ribosome. So we are going to take a minute and modify the ends of our pre-mRNA molecules. So we'll go back here for a minute. On your messenger RNA, I'm going to move this. When you look at the ends, I didn't label this for you, but you can go through and take a look at the sugars and the carbons labeled in that sugar. Remember the phosphate group that hangs off the end. The phosphate group is always attached to that 5' sugar. So the end where you have a phosphate hanging off is the 5' end of your RNA. And so remember the 5' end is going to receive that GTP cap. So this piece right here is going to bond, I'm just grabbing a scissors here, is going to bond to the 5' end of this. I'm going to cut that a little bit closer here. And we are just going to tag that right on like this. So here is our 5' cap. You can take a little piece of tape and tape that right on the end of your messenger RNA. The other end, slide that down real quick. The 3' end receives a poly A tail. Now, hopefully it's obvious to you, I'm not going to have you tape 250 more adenine bases on here, but we are going to add several. Take a minute and just tape on maybe half a dozen extra adenine bases to represent that poly A tail that hangs off the 3' end of your messenger RNA. So it should look kind of like that. Just add another half dozen or so adenine nucleotides. Tape those together. So those modifications, remember, help our messenger RNA to exit the nucleus and not get broken down before it is translated. So now our messenger RNA, sorry, we'll go back here for a sec. So now our messenger RNA is almost ready to go. There's one other thing that needs to happen. We're not going to model it in this activity, but there's one other thing that happens to messenger RNA before it exits the nucleus. And this, you can kind of tell this has to happen if we look at some numbers here. Cut all this part out. I don't even need all that. So now we have modified the ends of our messenger RNA so that it can leave the nucleus and not get broken down, but it's still not quite ready. Before we talk about what else happens to it, here are a couple of interesting facts for you to look at. The human genome contains between 19,000 and 20,000 genes. But the human proteome, which is the word for all the proteins in human beings, has been estimated to range up to several billion proteins. So it should be pretty obvious that we can't just have one gene that codes for one protein. That's not going to work. We don't have enough genes. And here's one of the ways that that works, that we can code for all these billions of proteins with only about 20,000 genes. One other process that happens before an RNA molecule leaves the nucleus is called RNA splicing, which means it gets cut and parts of it get glued together. So the final messenger RNA that leaves the nucleus is not as long as the primary transcript that was made. The sequence of nucleotides that codes for the actual polypeptide is usually not just a continuous row. The useless information has to be cut out, and the rest is kind of glued back together. The introns are the parts that are in between. They're things that do not code for information. The part that is expressed are the exons. So think exons are what exit the nucleus and then are expressed in the protein. And the cool thing about this process of splicing is that it can happen differently in different cells. There are spliceosomes. These are complexes of proteins and RNA that do this process. And the interesting thing is that different, one gene can end up giving rise to multiple types of polypeptides depending which segments are cut out. Imagine this. If I told you all, don't do this, first of all, do not do this. We are not modeling RNA splicing as part of this activity. But if I told each of you right now to cut out 12 nucleotides from your messenger RNA, but I didn't tell you which ones and I didn't tell you where, everybody would cut out a little bit different section, right? And then tape them back together. And you can imagine then the proteins that we made at the end would not all be the same. Your cells do that on purpose. They take this original pre-mRNA up at the top and they can splice it differently. In this path on the left, they spliced out the green segment. That was an intron that they got rid of. They left the red, yellow, blue. Those were the exons. And they produced this protein. On the right, they used that same pre-mRNA, the same gene, but this time they spliced out the yellow one. They left the red, green, and blue exons, and you ended up with this protein. So we get two different proteins built from the same piece of genetic information. This is a way that we can use the same gene in multiple different ways to produce several different proteins. So it's a pretty cool process that allows our bodies to produce way more proteins than we have genes through this process of alternative RNA splicing. Okay, so now Our messenger RNA is finished. Like I said, we are not going to model RNA splicing. Our pre-mRNA, the ends have been modified. We can pretend that splicing happened, but now it is ready to travel out of the nucleus to a ribosome. So at this point, you can take your DNA away because when the messenger RNA leaves the nucleus, it no longer interacts with the DNA. The DNA is finished. And it travels out to a ribosome in the cell. A ribosome is made up of two different parts, two different subunits. There's one called a large subunit and one called a small subunit. Very difficult to remember names. Each subunit is made up of proteins and one or more ribosomal RNAs. So there's another type of RNA that actually makes up part of the ribosome. And we'll talk a little more about what its role is tomorrow. The subunits are built in the nucleolus. Notice that O in there. This is not just the nucleus. The nucleolus is an organelle inside the nucleus that builds the small subunit and large subunit of the ribosome and then sends them out to float around in the cytosol. And they only join together to form a functional ribosome, like on the right here, when it's attached to a messenger RNA molecule. Otherwise, these two pieces separate and float separately around the cell. And so the messenger RNA travels out to a ribosome. The small and large subunit come together. And the other molecule that's very important at the ribosome in the process of translation, which we'll talk more about tomorrow, is the transfer RNA. So this is the third form of RNA that's involved in actually building a protein. Transfer RNA's job is to read the message in the messenger RNA and bring in the correct amino acid to build that polypeptide chain. And like I said, we're going to model translation tomorrow, so we'll use transfer RNA then. But transfer RNAs are also an RNA molecule. It's a single strand. If you look at these pictures up here, there's a few different views of what it could look like, but it's a single strand of RNA that's about 80 nucleotides long. It folds back on itself, and these nucleotides in between here kind of join with hydrogen bonds to make this kind of cloverleaf shape. At the very end, at the top of this picture, it forms an attachment site for an amino acid. So this is where the amino acid is going to bond to the transfer RNA. The other end, the loop down at the bottom, you can see has three bases kind of hanging off the bottom. That is called the anticodon. And we usually write that from 3' to 5' because it base pairs using those complementary base pairing rules to a codon, a set of 3 nucleotides on the messenger RNA, which we usually write from 5' to 3'. And again, we will be practicing this as we do translation tomorrow. But the anticodon here bonds to a 3 nucleotide codon on the messenger RNA. That bonding is very specific and has to be done correctly so that the correct amino acid is brought and the correct polypeptide is built. The top end carries an amino acid. And so in order to pick up its amino acid, each transfer RNA floats around out in the cytosol and interacts with an enzyme. There is a whole group of these enzymes called amino acid tRNA synthetases. This is a mouthful of a name, but again, if we break it down, ASE means it's an enzyme. Synthetase, to synthesize something, means to make or build, so it's putting things together. And in this case, it's putting together an amino acid with its appropriate transfer RNA. So the name says what it does. It's just kind of a mouthful. So these enzymes, there are about 20 different kinds. Each of them can bind to its amino acid. So they're very specific in this top binding site. Only one amino acid will fit there. And it can also bond with any transfer RNA that has an anticodon that codes for that particular amino acid. This bonding also has to be correct, the matching up of the transfer RNA with the correct amino acid. This has to happen correctly along with the anticodon bonding with the codon on the messenger RNA to ensure accurate translation of that genetic message. So from the messenger to the transfer RNA and from the transfer RNA to the amino acid, both need to be correct. So the last thing for today, and then we're going to prepare a couple of things to actually do this. Ribosomes and transfer RNA work together to do the process of translation and actually build the polypeptide. A ribosome is big enough to hold 3 transfer RNAs at the same time. So it has these 3 bonding sites, and they each have a specific name and a specific job. The middle one is called the P site. It holds the transfer RNA that's bonded to the polypeptide chain that's being built, okay? The A site holds the transfer RNA carrying the next amino acid that will be added to the chain. So you can think of A for amino acid, or A for adding, or A for arrival. That's where it first comes in. That amino acid gets added to the polypeptide chain, and then the ribosome slides down so that the previous tRNA is now in the E site, which is where it exits the ribosome. So what we are going to do to wrap up today and prepare for translation tomorrow, there are 2 things. First of all, you need to design yourself a ribosome. So this is where you will need some extra paper. Doesn't really matter what shape or what color or shape or anything like that. But your ribosome does need to have these 3, this E, P, A. I don't know if this will help you, but I remember Environmental Protection Agency, and then I never forget what order they go in. You need to have those 3 bonding sites, and you need a large and small subunit. You also need to cut out a stack of about 15 transfer RNAs to use tomorrow when we do some translation. So, my ribosome looks something like this. As you make your ribosome, so here's my large and my small subunit, you do need to mark and label your 3 bonding sites. As you do that, you can use your messenger RNA to help you kind of judge. Remember, a transfer RNA contains an anticodon of 3 bases. So you want to make it kind of big enough. Each of your bonding sites should be big enough to fit 3 bases. So that just gives you an idea of roughly how big to make your ribosome, so it will actually be functional in your model. The small subunit doesn't really need to have anything on it. If you want to label it small subunit, you are welcome to do that, but that's not required. In your packet, you have a couple sheets of transfer RNAs that look like this. You also need to cut out about 15 of those like this so that they'll be ready for tomorrow. You can leave them blank. We are going to fill in the amino acid name and the bases on each of these as we go tomorrow, but you should have them cut out before we start. Please have those 2 tasks taken care of before before Monday, so that we can continue with the process of translation then. Contact me if you have any questions. I hope you have a great rest of your day.